Hello my friends and welcome to another Cynic video. Here we're going to be looking at the infection and response topic, and with it we're going to look at how communicable diseases can be passed on. We're going to look at different types of disease. We'll look at how the human defense systems work defending us from infection. We'll look at what a vaccination is, how it works, the working and action of antibiotics and painkillers, how we've discovered and developed drugs, what monoclonal antibodies are, which is biology higher tier only, and what plant diseases are. So the way that we can define a communicable disease is something that can be passed from person to person. It's an infectious disease. And these infectious diseases can be caused by something called a pathogen. Now they are any disease-causing microorganism. So a virus, a bacteria, a protist, or a fungus. And these pathogens can be spread, they can infect plants or animals, but they can spread by either direct contact, water, or air. Those are the only methods we really need to know. However, when we look at types of disease later, you'll see that there are things like gonorrhea, which is passed through sexual contact. So there are other methods, but that's the ones that the examiners will expect us to know. And the reasons why these cause us harm, or why they make us feel ill, is because they usually infect our body. They get inside our blood or our tissue fluid, and they start reproducing rapidly. And as a result of that, bacteria will make toxins or poisons that damage tissues and make us feel ill, whereas viruses will live and reproduce in our cells. They use our cells for their own benefit. They use our cells' DNA and ribosomes to make their own proteins, and they will make so many copies in the cells until the cells actually burst and causes cell damage to them, which causes our cells to eventually die and the viruses to replicate. So there are 4 categories of pathogen, and we need to know some diseases from each type. The first type is viral diseases we're going to look at, and there are 3 types of disease within this category. The first one is measles, and measles is a disease which is becoming more prevalent again because people are avoiding having the measles, the MMR vaccine. It's a discussion for another day, but it leads to fever and red skin, eventually can be fatal, especially to those more vulnerable and with their immune systems, so the elderly and the very young, and obviously the very poorly as well. But these basically are spread from droplets through sneezes and coughs, and it can be really nasty. HIV is the second one, which can lead to AIDS. So HIV is the virus. This is the virus, that's what it looks like here. What it does is it attacks the immune system, specifically white blood cells. It's spread by sexual contact or exchange of bodily fluids, so blood from sharing needles. But what it will do is it will cause damage to the immune system, stop it from working, and it will lead to the autoimmune deficiency syndrome. So your immune system basically becomes deficient. It gets broken down, it's so destroyed that it won't work, and as a result you can pick up even the smallest of colds and it will cause you to get really poorly. So it's a very horrible disease. The third one is tobacco mosaic virus, which, as you can tell, tobacco, it affects only plants. In this case, it's plants including tomatoes, obviously tobacco, and basically the leaves will become discoloured, and as a result they cannot photosynthesize because we need that coloration. We need the green chlorophyll to be absorbing the colours that obviously are not green, so that photosynthesis can work. And if it's discoloured, it's not absorbing the right colours, therefore it can't photosynthesize as well. If it's not photosynthesizing as well as normal, it's not producing the key glucose, and without as much glucose, it's leading to less growth. The second category of disease is fungal. There's only one disease here that we need to know about which affects plants, and it's called rose black spot. Obviously affecting roses, and it gives them a kind of black spot appearance. It's often kind of discoloration of the leaves as well, so the same idea with TMV, but they stop functioning properly. We end up with less photosynthesis, we end up with less growth. It's spread by wind or water, this one, and we can treat it quite easily using fungicide, which specifically targets fungi, kills off the fungi. We have similar drugs that we can take as humans, and we could also remove and destroy the affected leaves from a plant, so a close monitoring of roses to make sure that they don't pick this up. In the bacterial category, we have salmonella, which causes it's from food poisoning, usually because of the unhygienic food preparation or not thorough food preparation or thorough cooking. Commonly comes from poultry, things like chicken, and found on eggs as well, and we can actually vaccinate poultry to prevent the spread or reduce the spread, so they don't then actually carry it themselves and pass it on to humans. It leads to fever and abdominal cramps, which lead to diarrhea and vomiting, but that's all because of the toxins released by bacteria. And the second type is gonorrhea, which is a sexually transmitted disease. It causes a thick yellow or green discharge from the vagina or penis and pain when urinating, and it's treated by use of antibiotics. And it's easily prevented using barrier contraceptives like a condom or a diaphragm, or something that prevents the contact of bodily fluids. And then the protist category, which is the fourth one, if you like, the extra final one that most people don't really know about. And in protists, there is a group of malaria protists. And these malaria protists use mosquitoes as a vector. And a vector is something like a vehicle, it's a carrier. So they use the mosquitoes as a carrier at a stage in their life cycle. And when malaria is picked up by mosquitoes and mosquitoes feed on humans, the malaria protist can get carried across into humans, and in humans what it will do is cause them to get a fever, which can be fatal, and the spread can be controlled by controlling the vectors. If we can control the mosquitoes, people won't get bitten by the mosquitoes that carry malaria, so by stopping them from breeding and by using mosquito nets, we can try and reduce the frequency of bites from mosquitoes that carry malaria. So let's look at how humans can defend themselves. So first off, we have a first line of defense to stop them getting into our body in the first place. And these are non-specific, which means they don't target a specific type of infection. They don't prevent a specific type. They're just trying to prevent all types of infection. So for example, our skin is a brilliant barrier. Our skin is just a physical barrier to our blood. It scabs over, things can't absorb through it, it won't let things through into our blood. In our nose we have hairs and we have mucus. Both of those are there to try and trap bacteria, stop them from getting into our lungs. In our trachea and our bronchi we have cilia and we have more mucus. So the trachea is the windpipe, if you like, and the bronchi are the branches from the trachea that go to the lungs. And in those, the cilia are tiny little hairs that waft mucus up to the top of your throat, so you swallow it, which goes into your stomach. And the stomach has acid, which hopefully kills off the majority of the pathogens that would get into your stomach. Some obviously make their way through. They're pretty sneaky. But there are obviously different ways that infections can get into your body, so things like cuts as well. And we're in this constant battle of evolution of our pathogens trying to get better at infecting us. As a second line of defense, if these pathogens get into our blood, we have white blood cells, which can do three things. So the first thing they can do is phagocytosis, which means engulfing. So I've got a Pac-Man here, he's going to go and chomp down on some bacteria. Basically what he does is he absorbs it, and we use enzymes to digest it. They're quite large white blood cells that do that, but that's the major way of destroying bacteria and pathogens in our body. If it's a bacterial cell or something like that, they will engulf it and break it down. But what they'll also do, which is very important, is antibody production. So they'll produce antibodies, which are kind of Y-shaped structures usually, and they have this region on them which is specific to the shape of an antigen that they bind to. So here we've got a bacterial cell. On the outside it's got these shapes which are made of protein, and they're called antigens, and they're kind of the markers that tell us what the cell is. So our own cells have thousands of these, and they tell our body that there are our own cells, and they'll say this is a liver cell, this is a retinal cell. But when we get invaded by pathogens, each pathogen has its own set of markers, these antigens. And what our white blood cells will do is over time they'll start to be able to produce white blood cells that fit to these. So the shape will be very specific, but they will fit to each of these antigens. And by binding to the antigen, they will be able to clump these bacterial cells together. They'll be able to make it easier to spot for the white blood cells that do phagocytosis, and basically lead to their inevitable destruction. I always think it's a bit like a laser targeting system. It's saying this is something that we need to destroy, and it is what ultimately will lead to you getting better. The antibodies speed up the process of the destruction of these cells, and if we can help to make antibodies work better by or faster, for example, by vaccination, then it'll be even better. They'll also produce antitoxins, which anti means stopping or going against, working against, and this obviously works against toxins, so trying to prevent the toxins from being able to work. So how can we do vaccination to encourage our body to defend itself even better? Well, what we do is we introduce small quantities of dead or inactive forms of a pathogen. So these won't cause you any harm. The side effects you might get from vaccination are because of your immune system working, which are what cause symptoms of a cold, for example, too. And what will happen is the white blood cells will produce antibodies. They'll have a normal reaction to these foreign chunks of or parts of pathogens inside your body, because they will have any reaction like that to any foreign body that isn't your own cell. And what they will do is they produce antibodies, and they'll leave behind these things called memory cells. And these memory cells will recognize the specific antigens that have been targeted previously. And with a second infection, the white blood cells that are memory cells are able to respond more quickly by producing a greater number of antibodies. So you get many of these made much faster. So whereas normally it could take a week, more than a week, to between 2 and 4 weeks really to have a full immune response to an infection, on a secondary infection we're talking more a matter of a couple of days. So there's really not even a chance for symptoms to start showing themselves. And if you think of a graph, we've got a graph here of the number of antibodies for a particular pathogen compared to time. At a first infection, we've got this point here, there's going to be 0 antibodies in your body for that pathogen, and obviously time is 0. So we're going to be starting down here, but as time goes on, our body is able to start producing antibodies until the point at which the infection's been cleared. The antibody numbers will then drop off because you don't need them anymore. So the level starts coming back down again. It doesn't go completely to 0 because, like the memory cells, need to be some left behind in your blood to fight a future infection. If then a future infection shows, just changing colour, an actual infection is found, so you've been vaccinated against measles. At this point, you have an immune response, you've produced a large number of antibodies, you've destroyed the infection, and then you're given measles for real. At this point, your body is able to produce antibodies much faster and in a much higher quantity, so that you can fight the infection and clear it and not even show any symptoms. It's wonderfully simple. If you had got measles the first time in this position here, you'd have the same system. So the first time you got measles, you would take at some point between 2 and 4 weeks to actually start fighting the infection properly, in which time you would have had serious fever, you would have possibly had a chance of dying from it. But by having a vaccination, you're responding to it much faster, you're killing it off before it can even affect your body in a negative way. Antibiotics and painkillers can be taken as a kind of drug form, or intravenously too, and antibiotics will actually treat the pathogen. They will try and destroy the pathogen. However, they'll only work for bacteria. So only work for bacteria. They will not work for viruses, they will not work for fungi, they will not work for protists, they only work for bacteria. The issue at the moment that we're facing though, and you can link this in to paper 2 in evolution, is where we're facing bacterial resistance, and in fact you need to know a little bit about it in paper 2, which is where we're misusing antibiotics, and so bacteria are being faced with antibiotics, and some of them are able to survive because of misuse, and these ones are able to pass on that ability to be resistant to antibiotics to their neighbours and through reproduction, and we're facing resistant strains, which is not good. And painkillers, however, only treat the symptoms. They do not destroy pathogens. So they will make you feel better, but they will not make you get better. Antiviral drugs, just a side note, this is also something that you need to be aware of. Antiviral drugs can be made, but they are difficult to develop because often when we make them and we find the mechanism of how they work, they end up damaging the body's own tissues as well. How do we discover, how do we find drugs, how do we develop and use them? So we're going to look at discovery first, and we'll talk about how we test and develop them a little bit later. So first up, we traditionally have found them from plants and from microorganisms, and the examples that we need to know are the heart drug digitalis, which was found from the foxglove. We found the painkiller aspirin from the willow tree, and penicillin, the one that you're probably aware of already, is from penicillium mold found on bread by Alexander Fleming. And those were all found in nature. We then extracted them, purified them, produced them in higher quantities, and we use them in humans now. Phenomenal, really. But most new drugs now are synthesized in the pharmaceutical industry, so they're made in a lab. However, the start point for that synthesis may come from a plant. Now, in the history of use of drugs, some drugs have been given to patients before they've really been trialled, before they've been tested to see if they work, whether they are actually toxic for humans, and tested to find out the appropriate dose to give people. So now we have a very rigorous system for making sure that they are safe to give out. And the system is to go through preclinical trials and clinical trials. And in preclinical trials, we'll test it on cells of humans, which is first just to see what effect it has on them. We can study their chemistry. We can look and see if they physically survive being exposed to drugs. We can then give it to tissues, which again is typically human tissues in a lab, not on an actual live human, and see what response they have to it. And we can then finally test it on animals, and we'll kind of go through a graded system of that. So we'll start off with animals like mice, and we'll typically then work on animals that are more similarly related to humans, like chimpanzees. But once we've tested for those, we're looking for, again, the toxicity, we're looking for efficacy, which is whether they work, and the dosage, as in how much we need to give them. If it passes those trials, and that can take a few years to do, we then start clinical trials. And in clinical trials, we're working and testing on humans. And so the first test that we actually do isn't on patients who are suffering from the problem that the drug is trying to treat. We actually test on healthy volunteers, so very normal people, a wide spread of them preferably, so that we're testing a bunch of age and gender and race and everybody really. And the idea of this is to look at toxicity. And we'll compare a bunch of people who are given a placebo, which is it's like the drug. The method of the administering of the drug is the same, but they aren't actually being given a drug. They're given just the equivalent of a Smartie. It's nothing. And then there'll be another group of people that are given the drug, and we're looking for any difference in how they respond. So if one starts being violently sick with the actual drug, then obviously we know the drug is causing that and it's toxic. If it passes that phase, we'll then enter a test on a small group of patients. And in the small group of patients, we're testing for whether it works, again, in proper humans now, and we're also testing the right appropriate dosage. So what dose do we need to give to them to make it work? And can we kind of limit the dose that we give so it doesn't cause too many side effects? And if it passes that trial, we'll then test on a very large group of patients. So we're talking thousands of people, again, on a large scale, looking now at toxicity. We're looking for those 1 in 1,000 outcomes, 1 in a million outcomes, the very rare things that can occur. We're also looking for efficacy to confirm in a large statistical group of people to see if it works. And we're also looking for dosage. But pretty typically, all three of these stages are following the double-blind trial procedure, where some patients are given a placebo, and neither the patient nor the administering doctors or nurses know who is receiving the placebo or the actual drug. It's to prevent bias. We don't want any bias at all. We don't want the doctors to be giving out the real drug saying, you'll feel better soon, subconsciously without even realising it. So removing knowledge of who is being given the drug and the placebo eliminates that bias. One of the great focuses of biology at the moment is the study of monoclonal antibodies and trying to use them in healthcare. And these, it's a biology higher tier part only, so very niche part of the topic. But in this, what these monoclonal antibodies are a clone of one type of antibody. So we take a single clone of the cells that are producing them, and these cells that are producing antibodies all produce the same ones. They're clones of each other, and so all the antibodies that are produced are identical and specific to only one binding site on one protein antigen. So we spoke earlier about antigens. These will only bind to one of them, and we can produce millions and millions of these identical antibodies that do a particular job. So they can be created to target a specific chemical or specific cells in the body, or anything that we want to, really. So these are particularly cool, really useful, because we can stick stuff to the other end of the antibody. So we've got the part that does the binding. At the other end, we can stick radioactive substances to do radiotherapy. We can put dyes, we can put chemicals. We've got a wonderfully creative part of biology here that we can work with. So how do we produce them? Well, the first phase is to actually work with a mouse, and we stimulate the mouse to make a particular antibody. So we might inject the mouse with some antigens, but generally trying to get an immune response. We'll then take the white blood cells from the mouse by taking actually out its spleen, and from those we take out lymphocytes, which are the cells that produce antibodies. These are actually B lymphocytes. And these B lymphocytes we then combine with a tumour cell, because tumour cells reproduce rapidly. They have no reproduction limit, so they can reproduce as many times as they like, whereas typical cells kind of stop reproducing after a while. So we've got these quick reproducing cells that can also produce antibodies. Brilliant combination. So these cells then will clone via mitosis, a process you know from topic 1, and they'll make identical cells and therefore identical antibodies. They'll produce loads of these antibodies in a lab, and these antibodies can be collected and purified. So we need to know a handful of uses of monoclonal antibodies, and the first one is in diagnosis. So, for example, we can use it in pregnancy tests. There's a lovely question in one of the past papers on pregnancy tests where you're given how the pregnancy test works, and you need to just explain it using the diagram that you provided. We also can use it in labs, so we can use it to measure hormone or chemical levels in the blood or to detect pathogens. We can also use it by binding dye to the antibody to locate or identify specific molecules in a cell or a tissue, so basically by doing chemical testing. And we can also use it to treat diseases. So if we stick the antibody to a radioactive material or toxic drug or chemical, it can then bind to the specific cells or types of pathogen or whatever it is that we're trying to target. So if we know the antigens on the cancer cell, we can produce monoclonal antibodies to target that antigen, and therefore we'll only kill off the cells that have that antigen, just eliminating the cancer. It'd be phenomenal if we could get it to work properly. But there are some problems. At the moment, they create a lot more side effects than we were expecting, so they're not as widely used as first thought, but loads of research is still going on into monoclonal antibodies in medicine. And the final part of this topic, we need to know a little bit about plant diseases, and this is biology only, so not combined science. And in this we need to know firstly, for foundation and higher tier, that a nutrient deficiency is a type of disease, and we need to just know about 2 types of deficiency, and that is it. So we need to know about nitrates and magnesium deficiencies, which is just where they don't have enough of it. So if they don't have enough nitrates, nitrates are used to make protein. If we don't have enough nitrate, they end up with stunted growth because there's not enough available for protein synthesis. And the magnesium is used to make chlorophyll, so if there's less magnesium, there's less chlorophyll produced. It leads to something called chlorosis, which means that the plant will have stunted growth because there is not as much photosynthesis going on, and therefore it can't grow as tall. And if it can't grow as tall, it's because it's not got enough glucose being produced. Now, if you're doing higher tier, we need to know 2 extra parts to this, which are very simple. So I don't understand why it's higher tier, but anyway, the first part is what we can look for in a plant to know that it's got disease. So we might see stunted growth or spots on a leaf, might see areas of decay on the plant, or growths, literally like tumours almost for plants. We might see malformed stems or leaves, they're not looking normal. They might be discoloured, there might be a presence of pests on them like aphids. Those are diseases, okay? They're signs of diseases. Then, if we know that they have a disease, because we recognise these things, we need to know what you could do to identify which disease that is. So again, this is really simple. You could reference a manual or a website that's going to tell you what the malformed stems indicate. We could take it to the lab to test for pathogens, or we could actually use a testing kit if you're in the field to test for a particular disease using monoclonal antibodies. Simple as that. So that brings us to the end of the topic. If you have any questions or queries or comments or suggestions, please do write them down on the comment section. Otherwise, do like and subscribe and check out the other videos for the rest of this topic.